The idea of reversing aging once belonged to the realm of science fiction, but recent advances in the field of epigenetics have brought this ambitious goal closer to reality. Unlike genetic mutations, which change the actual sequence of DNA, epigenetic modifications affect how genes are expressed, essentially turning genes on or off without altering the genetic code itself. Over time, these regulatory mechanisms become disorganized, contributing to age-related dysfunction and disease. Now, scientists are asking a revolutionary question. What if we could reprogram the epigenome to restore a more youthful, resilient state? Epigenetic aging is largely driven by factors like DNA methylation, histone modification, and chromatin remodeling, all of which influence which genes are active in a given cell at a given time. As we age, patterns of gene expression shift in ways that reduce the cell's ability to repair damage, respond to stress, or maintain tissue-specific identity. This epigenetic drift leads to the gradual decline in organ function and systemic health seen in aging, even when the underlying DNA remains intact. In essence, the software that runs our genetic hardware begins to degrade, and the result is cellular confusion, inflammation, and reduced regenerative capacity. Epigenetic reprogramming aims to reset this biological software. The most dramatic proof of concept comes from the use of Yamanaka factors, 4 transcription factors, October4th, SOX2, KLF4, and c-Myc, that can revert adult cells back to a pluripotent, embryonic-like state. While full reprogramming erases a cell's identity entirely, recent experiments have shown that partial reprogramming can restore youthful function without compromising the cell's identity. In mice, periodic expression of these factors has reversed markers of aging, improved tissue repair, and even extended lifespan in models of premature aging. The implications are staggering. Aging, once thought to be irreversible, might in fact be something that can be biologically reset. Crucially, this isn't about changing who we are at the genetic level. Our DNA remains constant, but the way it is interpreted by the cell is malleable. That makes epigenetic reprogramming not only powerful but potentially safer than gene editing, which carries risks of unintended mutations. Instead of altering the blueprint, researchers are adjusting how the blueprint is read, reactivating youthful gene networks, and silencing those associated with aging and decline. One of the most compelling examples of this is in eye health. In a groundbreaking study, researchers used epigenetic reprogramming to restore vision in aged mice with damaged optic nerves. The treated cells showed a dramatic reversal of age-related decline, with reactivation of youthful gene expression and regeneration of damaged neurons, something previously thought impossible. Similar strategies are being explored for the heart, liver, muscles, and brain, with early results pointing toward improved function, reduced inflammation, and enhanced resistance to cellular stress. Beyond lab experiments, scientists are also mapping epigenetic clocks, patterns of DNA methylation that correlate closely with biological age. These clocks are now being used to measure the effectiveness of longevity interventions, and in some cases, subtle reprogramming through diet, fasting, exercise, or pharmacological agents has been shown to reverse these clocks by several years. Compounds like metformin, NAD+ precursors, and natural polyphenol are believed to influence the epigenome indirectly by modifying how cells respond to stress and nutrient availability. The long-term goal is to develop safe, targeted therapies that can reset key aspects of epigenetic aging without the risks of full reprogramming. While challenges remain, especially in delivering epigenetic reprogramming factors precisely and safely in humans, the potential is enormous. It may be possible, within our lifetime, to restore aged tissues to a youthful state, regenerate damaged organs, and significantly delay or even reverse the onset of age-related diseases. This would mark a fundamental shift in medicine, from treating symptoms of aging to addressing its root causes. Epigenetic reprogramming offers a vision of aging that is flexible, not fixed. By learning to rewrite the instructions that govern cellular identity and repair, we may soon move beyond simply slowing aging toward actually reversing it, restoring resilience, vitality, and function in ways once thought unimaginable. It's not a fountain of youth in the mythical sense, but it might be something even better, a science-driven path to lasting cellular renewal.